Welcome everyone to Foresight's Biotech and Health Extension Strategy Group, sponsored by 100 Plus Capital. I will post in this chat 3 links. One is to all videos and summaries of our previous meetings. Bolek has written fantastic summaries for other people to read up for those who can't join us here. Then the second link I'm going to share is an application to our accelerator. We had at our last meeting already Jean-Albert on brain cell replacement. So if you're interested in the accelerator, please take a look at that meeting, at that keynote program, and reach out to me if you have any questions. And then lastly, I'll share our collaboration spreadsheet. And so this is just a reminder, fill yourself in the member registry. I do get a lot of folks always telling me how they actually do find people via that spreadsheet. So I think it's proven quite useful. Okay, great. So today we're going to be discussing clinical trials and senolytics with Dr. Kirkland from the Mayo Clinic. And I'm really, really happy he's joining us again. At the previous meeting that we had last year, where we had our first Q&A with him, the video got a comment where someone called you the most intelligent man he's ever seen. So no pressure for this video and for this meeting. Thank you so much for having joined us back then. It was a really, really fantastic talk. I tentatively named this session Clinical Trials and Senolytics Q&A, just because those were the 2 topics that unsurprisingly people thought were most engaging at our previous meeting, but feel free to discuss anything you'd like, and likewise for everyone else. As we start to collect questions, feel free to collect questions on other things that you may find inspiring from Dr. Kirkland's very extensive bio, which I'm going to share here in the chat. And yeah, I mean, you've not only really published, your lab hasn't only really published like one of the first papers on senolytics, but really since then you've paved the way in terms of overcoming regulatory hurdles for a variety of different interventions. So thank you so much for joining us again. I know the group really, really appreciates your advice. And without further ado, I think I'll just let you present for, I think, about 30 minutes. In the meantime, we'll be collecting questions, and then we'll start into a Q&A. So I'll be in the chat in case anyone has questions. And from my end, again, just thank you so much for joining us. And it's much appreciated by the group. All right.
Well, thank you very much for the kind introduction. And we're just groping our way along. And this, what I'm talking about is the work of a lot of people. It's not just me, and I'm not particularly smart. I just happen to have an office with a lot of smart people around me. So I'll try to share my screen. I did talk fairly recently on this program, so I'm just going to try to briefly recap what we discussed before, and then hopefully give a few updates. So we'll talk again about senolytic agents. And as we discussed the last time, and I think as many of your other speakers have pointed out, aging, chronologic age is by far the largest predictor for most of the chronic diseases that account for the bulk of morbidity, mortality, and health expenditures, and is also related in advanced chronologic ages to the geriatric syndromes, that's things like frailty and age-related muscle impairment and mild cognitive impairment, as well as phenotypes that we see in older individuals, that is how they look and their characteristics, and decreased physical resilience or decreased ability to recover, say after surgery or an infection. So these aging processes, of course, begin at the time of conception. Fundamental aging mechanisms can develop at sites of age-related diseases anywhere from early development on through later life. The process that we're interested in is cellular senescence, first described back in 1961 by Hayflick and Moorhead. And senescent cells are cells that have lost replicative potential and are essentially replicatively arrested, but they're viable. Some senescent cells are very metabolically active and have increased protein production. And they're hard to kill. They're resistant to apoptosis, to programmed cell death, and they're normally only removed by the immune system. There are no completely sensitive and specific markers for senescent cells. They accumulate at sites of age-related diseases and chronic diseases. They accumulate in healthier older individuals across multiple tissues in later life, but they can appear at any point in any vertebrate from conception on. Senescent cells, for example, in the placenta are responsible for producing factors that drive the baby through the birth canal. Senescent cells clear up tissue damage after wounding. They're important during fetal development, and they act as a defense against cancer. So senescent cells will form in response to stimuli that drive a cell into cancer. So some of oncogenes or repeated replication or DNA damage can make a cell become senescent. And if they appear in the middle of a cancer, they slow down the rate of growth of the cancer, but also some senescent cells, 30 to 70%, have a senescence-associated secretory phenotype where they produce factors that kill cells around them. So you don't want to interfere with the ability to generate senescent cells, but once they're there, many, if not most, senescent cells harbor cancerous mutations. And if you make them start dividing again, or if they escape their replicative arrest, they can come back as cancers. So, and once they...
Can I ask a quick question, please?
Sure.
So this is a lot of different functions and properties and benefits that are associated with these senescent cells, at least in some cases that you just went over. Is senescent really the right word for them? And how is it defined? Is it just defined that they've stopped replicating, and then beyond that, there's all these different possibilities for what they're doing, good or bad?
It's a cell fate like replication, differentiation, apoptosis, or necrosis. It's the fifth cell fate. So any cell can become senescent. They can become senescent in response to damage signals, repeated replication, problems with macromolecules, problems with intracellular organelles, in response to infectious antigens, and in response to drugs or radiation or other kinds of damaging insults. So any kind of cell can become senescent. Senescence is basically defined as essentially irreversible loss of capacity to divide. It takes 10 days to 6 weeks for a cell to become fully senescent, so it's slower than other cell fates. Some, but not all, senescent cells can have a secretory phenotype where they damage the cells around them, spread senescence, interfere with stem and progenitor cell function, damage tissues, and can produce a range of signals that attract, activate, and anchor immune cells. So this is a cell fate. It's a necessary process. You don't want to interfere with the ability of a cell to become senescent. But if they become senescent and they persist and they're not cleared by the immune system and they achieve a threshold above which they spread faster than they can be removed, they spread through their secretions faster than they can be removed by the immune system, then they start to take off and systemic problems can develop. There are probably two types of senescent cell at least. And the nature of senescent cells depends on the starting cell type, what induced senescence. There are 40 or 50 things that can do it, and how long the cell has been senescent. But 30 to 70% produce damaging factors. We call those deleterious senescent cells. And other senescent cells don't produce those damaging factors. We call them helper senescent cells. So those cells are important in wound healing and so forth. So the definition of senescence is basically entry of a cell into essentially cell cycle arrest that's irreversible unless there are further mutations or things that happen to that cell. So senescent cells have been found they're found at etiologic sites of multiple chronic diseases like in fat tissue and diabetes, or the pancreas and diabetes, the brain and Alzheimer's disease. They accumulate with advancing age. Their rate of accumulation is slowed by interventions such as caloric restriction that are associated with an increase in health span. That was published in 2004 by Ned Sharpless, who's now director of the National Cancer Institute. And that prompted us in 2004 to ask whether accumulation of senescent cells that have a SASP and are damaging, whether that is causal with respect to age-related phenotypes of geriatric syndromes and chronic diseases, or it's just associated. So we began trying to develop ways to clear senescent cells to ask if this was more than a causal relation, if this was more than an association, was in fact a causal relation. We eventually, after trying multiple ways to kill senescent cells, like using fusion proteins and high throughput library screens and so forth, remembered that senescent cells survive despite the fact that the senescent cells that are the deleterious ones with a secretory phenotype kill cells around them. So that led us to hypothesize that they must have survival pathways that defend them against the things they're using to kill cells around them. So we used bioinformatics approaches to look at proteomic and transcriptomic databases. We found indeed they have upregulated pro-survival pathways in a network that depends on the senescent cell type. And in some respects, the other hypothesis that we use to develop senolytic drugs is that in some respects, senescent cells are like cancer cells that don't divide. In fact, many of them are cancer cells before they've become dividing cancer cells. And those two hypotheses allowed us to use bioinformatics approaches and then other approaches to develop senolytic drugs. So we found there are indeed pro-survival networks. We found that disabling some or combinations of those networks would transiently, just for a few hours, would allow senescent cells that have a secretory phenotype to kill themselves instead of the cells around them to basically commit suicide. And that's how senolytics act. So because it takes 10 days to 6 weeks for new senescent cells to form, these agents can be given in a hit and run manner once or twice if there's an insult like radiation that's not repeated that has caused senescence, or in, say, a situation like high fat feeding and obesity and diabetes, periodically as new senescent cells form. So in those situations in experimental animals, the drugs can be given once every 2 weeks or once a month, and that is the same effect as giving these agents continuously. We're not trying to occupy a receptor or inhibit an enzyme. We're trying to get rid of a cell type. We're trying to allow these cells to kill themselves. A 2 to 3 hour exposure to these drugs is enough to do so. And then it takes 18 hours for the cells to kill themselves. So they can be given in a hit and run manner. So we moved away in developing these agents from the old-fashioned single drug, single target, single disease approach to using combinations of agents in some cases, or agents that act through multiple mechanisms to transiently disable these networks. We're not going after a single target, we're going after a network of pro-survival pathways that sometimes are redundant. And the view is that if the geroscience hypothesis is true, we might affect multiple age-related disorders, chronic diseases, and so forth that are associated with aging. So the strategy for developing these agents was much closer to developing antibiotics and drugs with a single molecular target or for a single disease. And there are multiple, I won't go through it all, I went through it the last time, animal models of various diseases and conditions in which intermittent senolytic administration appears to alleviate some of those conditions or delay or prevent them. One of the things that we have is something called the Translational Geroscience Network. It's a consortium of the 9 organizations that I've listed there. It's funded by the National Institute on Aging. And this network is there in order to help take drugs from the bench to the bedside in early phase clinical trials. And one of the things that we have through this network is what we call a facility for geroscience analysis, where we can study samples, say blood or urine or cheek swabs that contain cells from across these clinical trials. The clinical trials are not just of senolytics, but they're also of other agents that target fundamental aging processes. And eventually what we'd like to do is combine some of these interventions to see if they're less than additive, or synergistic. And we also want to begin doing trials where we combine these interventions with lifestyle interventions and also with disease-specific drugs, given the situation that fundamental aging processes may be root cause contributors to serious diseases, but in some cases there are some disease-specific drugs. And the question would be, does combining these approaches result in a synergistic, greatly improved response over using just a drug that targets a fundamental aging process alone versus targets a disease alone? So in that network, and there are other clinical trials underway of senolytics, in that network, there are currently 15 clinical trials of senolytics. As I mentioned, the network is not only studying senolytics, there are clinical trials of other kinds of agents, including drugs related to rapamycin, metformin, and a list of others that target the pillars of aging. And so I'll go through a few of these trials. None of them, there are only a couple of early results from a couple of the trials, so we've got a long way to go before we know that these drugs are safe and effective in humans. The earliest drugs we chose to bring to trials are dasatinib, which has been on the market since 2006. It's used as, and any physician can prescribe it in the US at least, for treating various kinds of cancers like lymphomas and chronic lymphocytic leukemia. It's also used off-label to treat a number of skin conditions like scleroderma. It's a tyrosine kinase inhibitor, but it's not like other tyrosine kinase inhibitors. It inhibits a particular tyrosine kinase that some kinds, but not all kinds of senescent cells depend on, dependence receptor tyrosine kinases, which are Src kinases. Another agent we've been looking at is quercetin. It's in apple peels. It's what makes them taste bitter. It will target other kinds of senescent cells that dasatinib may not target very well. And the combination seems to be synergistic. And furthermore, quercetin reduces the side effects, potential side effects of dasatinib. The other agent that we're looking at in early trials is fisetin, and in some of the trials we're comparing dasatinib and quercetin on the one hand to fisetin on the other. Fisetin is also, it's very closely related to quercetin. It's one hydroxyl group different. It's one of the three naturally occurring flavonoids that appears to be senolytic. It's present in strawberries, for example. But we have to give very, very high doses. You'd have to eat 15 pounds of strawberries in 5 minutes to get the kind of dose we're giving to act as a senolytic in a hit and run manner. We chose these agents because their side effect profiles are relatively well known, and they've got short elimination half-lives because we're trying to use agents where we can briefly expose to the drugs, and then we want them out of the system because what we're trying to do is kill senescent cells, as I mentioned. They take 10 days to 6 weeks to come back. We're not trying to continuously occupy a receptor or inhibit an enzyme. So we're not looking for steady state levels of these drugs in the blood. We're trying to use them in a hit and run manner. So a couple of the trials are in frail elderly subjects, the AFFIRM trials. These are of fisetin. They're placebo-controlled. They're both about halfway through now. One of them is for elderly women with extreme frailty with a gait speed of less than 0.6 meters per second. That incidentally carries about a 2-year 50% survival. So having a slow gait speed and frailty, if you're elderly below 0.6 meters per second, is probably a worse prognostic indicator than most cancers. There are several trials just beginning, and there was a pilot, a couple of pilot trials done before these trials began for Alzheimer's disease. One of them is Alzin Light that just is beginning to recruit now. And that is to look at targeting, what we call target engagement. Are we actually killing senescent cells in these subjects? I'll explain that trial a bit more in a moment. There are there's a multi-center double-blind placebo-controlled trial that's about to begin called STOMP-AD. There are trials for chronic kidney disease. There's a trial looking at inflammation and stem cells in diabetes, and preliminary results from that were published, which I'll show in a moment, but it's continuing. There are trials in bone marrow transplant recipients. This trial is almost finished. So people who get a bone marrow transplant get a high dose of chemotherapy and radiation that can induce, unfortunately, senescence as a side effect. before they get their bone marrow transplant, because you have to you have to destroy basically their own immune system before they can get a bone marrow transplant, for example, for various cancers. Turns out that a number of people who've had bone marrow transplants, not all of them, but some of them after 3 to 5 years get an accelerated aging-like state. And so that led us to test whether clearing senescent cells would alleviate that accelerated aging-like state where these people get cognitive impairment, diabetes, second unrelated cancers, and atherosclerosis with heart attacks and strokes. So some of them sort of look like they're aging very rapidly. There is a trial based on that same kind of logic at St. Jude that's been funded by the National Cancer Institute. And that trial is comparing fisetin to dasatinib and quercetin to a placebo. It's an open label trial though, but it's randomized. That trial should be beginning in June. It's funded, approved. And what it's to do is to look at the accelerated aging-like state that can develop in people when they're in their 30s and 40s who've been treated for cancers as children. So people who've been treated for leukemias or lymphomas as children have chemotherapy and radiation that can induce cellular senescence. And I'll show you the data confirming that, and I'll discuss that trial in a bit more detail. There's a trial about to begin, double-blind trial, in idiopathic pulmonary fibrosis. Preliminary results from an open label arm of that were published. And there's another safety trial which is about to be published. But these were a lead up to a larger double-blind placebo-controlled trial. And I'll come back to that in a moment. There is a trial which I'll explain in a bit more detail to look at age-related osteoporosis, which is about halfway through now. It's recruited about slightly more than half the subjects. That's an NIH-funded trial. There's a trial funded by the Office of Naval Research to look at osteoarthritis, which is a disorder linked to senescent cell accumulation in and around the knee joints. There's several trials that are underway or beginning for coronavirus and its complications. There's an outpatient trial, a hospital trial, and a large nursing home trial that's funded by the NIH that is just beginning now. So the idiopathic pulmonary fibrosis trial I mentioned, idiopathic pulmonary fibrosis is a usually fatal lung disease that occurs in older subjects. It's associated with senescent cell accumulation in the lungs. In animal models of this particular condition, where a chemotherapy agent is inhaled by mice, these drugs appear to alleviate the condition. So a clinical trial, an open label, non-placebo controlled clinical trial was done where 9 doses of dasatinib and quercetin were given over a 3-week period. And then 5 days after the last dose, a number of frailty measures were made, because we know in experimental animals and people with idiopathic pulmonary fibrosis-like states that frailty is a key parameter that you see. And in the animal models, this is alleviated faster than the pulmonary fibrosis, which takes quite a bit of time to alleviate with senolytics in animal models. So the decision was made to look at frailty in these subjects. And there appeared to be within subjects before versus after treatment, an improvement in 6-minute walk distance, gait speed, ability to stand from a chair, and the short physical performance battery. But I'd strongly caution that there are learning effects for all these tests. This was not placebo controlled, it was not blinded. It's very early, and the entire reason for doing this was only to know if there was some degree of justification in going on to a double-blind placebo-controlled phase 2 kind of trial. So that's the only reason this trial was done. There was a trial done, as I mentioned before, an early phase of the study in obesity and diabetes. In obesity and diabetes, a lot of senescent cells accumulate in adipose tissue and fat tissue. So in these subjects, a biopsy was done of their subcutaneous fat at day 0 and another biopsy at day 14. On days 1, 2, and 3, they got doses of dasatinib and quercetin by mouth. Then they didn't get any more drug after that. These drugs are cleared from the body within 36 hours because the elimination half-life of dasatinib is 3 or 4 hours, and of quercetin is about 11 hours. So the drugs are long gone by day 14. So in comparing the day 14 to the day 0 biopsy, there was a decrease in some markers of senescence. There are no perfect, completely sensitive and specific markers of cellular senescence. So we normally have to measure several different markers to convince ourselves there may be an effect on senescence. It's, you know, a question was asked about how do you define a senescent cell, and it is very difficult to define them at the level of markers. Also, immune cell infiltration. What happens in these people is with obesity and diabetes is they get inflammation of their adipose tissue with infiltration of things like macrophages, immune cells that cause inflammation, CD68 cells. The abundance of these cells decreased in their adipose tissue. We know from other studies that senolytics do not kill these cells. What they do is attract, activate, and anchor these kinds of immune cells. So we know from experimental animal studies that that senolytics don't directly kill these cells, but they reduce their abundance in things like fat tissue and animal models of obesity by reducing the senescent cells there within the adipose tissue that are attracting, activating, and anchoring immune cells. So in these human subjects, we found decreased activated macrophages at day 14 compared to day 0, and also less fibrous tissue in their fat tissue or crown-like structures. And there was a decrease in blood markers of senescence. Again, we can't follow any single marker. Through the Facility for Geroscience Analysis, we measure over 100 parameters in the blood to look at the pillars of aging. And many of those are to give us an idea of whether senescent cell burden is being affected or not. Here, a composite score is being looked at of several factors that can be produced by those senescent cells that cause harm through their secretory phenotype. And again, a 3-day course of treatment with these agents resulted in a decrease in these factors. So that gave us confidence to go on to the larger double-blind placebo-controlled trial. So I mentioned that there are no completely sensitive and specific markers of senescence. The Facility for Geroscience Analysis of the network, we measure well over 100 parameters, as I mentioned. Some of them are proteins that are in the blood. Some of them are things related to DNA and RNA that are in the blood or in cerebrospinal fluid or other body fluids. We measure a range of markers within cells. So we look for senescent cells in the blood, but we also look at epigenetic clocks and all kinds of other things. And we also look at other fundamental aging processes. It seems they're all interlinked. So if we target senescent cells, we tend to affect all of the other pillars of aging, as far as we know. And conversely, if we use agents that target other fundamental pillars of aging, we tend to affect senescent cells or their burden. So the Alzheimer's trial, as I mentioned, that's mainly a safety and feasibility trial. It's open label. It's looking at subjects for a few months before versus after starting intermittent senolytics. And this is in subjects with early but established Alzheimer's disease. And the purpose of this particular phase of the Alzheimer's studies is to know how big a placebo-controlled trial is going to have to be. So this involves subjects who are over 65. It's just beginning to enroll. They've got intermediate Alzheimer's disease as evident by cognitive function testing and looking at various markers, including PET imaging. It's going to be a small trial. It's mainly looking at target engagement, as I mentioned, and it's going to be of intermittent dasatinib and quercetin. And this is based on studies in animal models where there appeared to be some alleviation of the Alzheimer's-like state that some mouse models have, where we find that there are senescent cells in the parts of the brain that control memory and executive function, and that senolytics in those animal models improve new nerve production, reduce inflammation in the brain, improve blood flow in the brain, and decrease or even partially reverse brain shrinkage. So we're seeing if that's true in patients with Alzheimer's. In patients with Alzheimer's, we know they have senescent cells in the hippocampi of the brain and the frontal lobes that control respectively memory and cognitive function. So in that trial, the main thing we're looking for is safety and tolerability. We don't know if these drugs are safe. So these drugs are being given intermittently over a 90-day period. There are a lot of target engagement parameters being measured in their blood, their cerebrospinal fluid, and also looking at MRI and other kinds of imaging, as well as cognitive and functional status. There's a trial that I mentioned before is over halfway through, looking at senolytics for age-related osteoporosis. They appear to be effective there in animal models, where animal models of hormone deficiency-related osteoporosis, these drugs are not so effective. That seems to be, hormone deficiency-related osteoporosis seems to be more independent of fundamental aging processes than age-related osteoporosis. So in this trial, dasatinib and quercetin are being compared to fisetin. It's a placebo-controlled trial, and it'll be it's just over halfway through, and these subjects are getting 2 days of senolytics, 13 days off, 2 days of senolytics, 13 days off, over a number of cycles out to 140 days. So they're getting 7 cycles of these drugs. And again, this is based on preclinical animal model studies. And in addition to looking at bone mass, there are all the blood parameters that are being done in all the other trials are being done here as well as looking at bone turnover markers in the blood and imaging of bone. I mentioned before about the childhood cancer survivors, these people in their 30s and 40s who develop an accelerated aging-like state. At St. Jude, they follow all of their patients for life as much as they can. They've got over 800 of these subjects. And Kerry Ness and Greg Armstrong there found that senescent cells in the blood in these people who had received treatment for cancers as children are now in their 30s and 40s and are developing frailty, heart attacks, strokes, second cancers, Alzheimer's disease, and look like they're much older than they are. They found that the people with a slow gait speed and frailty had accumulation of senescent cells in their blood. So that led to the trial that the National Cancer Institute has funded in these childhood cancer survivors with difficulty with gait speed and other criteria of frailty. And it is like some of the other trials comparing dasatinib and quercetin to fisetin, and it's going to be 2 3-day courses over that are a month apart, with the main endpoint again being target safety and also target engagement, and then looking at gait speed. So that trial is funded. It's going to actually begin enrolling probably in June. We found with collaborators at Harvard that transplanting organs from old to young animals results in spread of senescence from the transplanted old organ to the young individual's own organs. And this isn't because of cells migrating out of the old organ. It's because senescence through its secretory state can cause the recipient's own cells to start becoming senescent because of the factors that senescent cells produce. So this just shows that if we transplant hearts from old to young mice, we start seeing senescent cells in the recipient's lymph nodes in their liver, and these are the recipient's own cells. And this is because, in particular, of something called mitochondrial DNA, which is released by senescent cells, but other factors as well. The transplant surgeons have known that organs from old donors don't work very well in recipients. This is part of the, an outcome of this is that in people who die, say in car crashes, who are over 50, surgeons are reluctant to use their kidneys, even if they've signed a donor card. And and they've got the kidneys. So we're throwing away 35,000 kidneys a year that could be used for transplantation because they're from older donors. So in these heart transplant animals, if we treat the donor, the heart being transplanted, or the recipient with senolytics, we're able to reduce the spread of senescence, and we're able to increase survival of the recipient. In fact, we're able to increase survival of the recipient to the same point that we see in recipients of hearts from younger individuals, and where we recreate the kind of transplant rejection treatment that is given to people, and plus or minus senolytics. So we actually get very, very good survival in these animal models. So that has led to early phase trials, taking kidneys that are being discarded because they're from older donors and asking if we can regenerate them so they could be used. Kidneys are put onto perfusion machines after they're removed from the deceased donor. And that allows us to measure senescent cell burden by looking at various parameters in the kidney while it's on the perfusion apparatus, and also to look at the function of the kidney. And we can administer senolytics to the kidney on the perfusion machine. We're doing a similar thing with the University of Groningen in North Holland, trying to rehabilitate liver transplants from older donors. The Fazetin trials for coronavirus, the one which is underway is in hospitalized patients who are admitted with coronavirus infection and of respiratory difficulty. And it's to test the efficacy of senolytics in alleviating respiratory dysfunction. We know that coronavirus activates the secretory phenotype of senescent cells and can also cause cells to become senescent. And in animal models of coronavirus, if we treat with young animals infected with a version of coronavirus don't die, older animals do, much like humans. And if we treat the older animals with senolytics, we prevent their dying, we reduce cytokine storm, and we improve antibody responses. So this trial is underway to see if we can recreate that in humans. It's just over halfway done now. But you know, it's a double-blind, placebo-controlled trial, so we don't know whether there are any positive results or not. The main thing we're looking for is side effects. We do know that we're not seeing much in the way of severe or serious side effects, but I lie awake every night worried about that. The trial includes people over 60 with a recently positive PCR for coronavirus, or 18 to 59 with risk factors, including things like obesity, diabetes, asthma, and so forth, which are also states in younger individuals that are linked to early accumulation of senescent cells. The protocol is a long one. We're trying to see if we can reduce not only respiratory difficulty and look at side effects and so forth. We're also trying to see if we have an impact on long hauler syndrome. There's a trial which is just beginning. It's going to be in, when it's fully rolled out, in 129 nursing homes across the United States. It's funded by the NIH. And this will be for patients who who test positive or have tested positive for coronavirus who are nursing home residents. We know that they have, that they don't do very well. So this trial is beginning in a small way at Mayo affiliated nursing homes, then it's going to spread across the Translational Geroscience Network. And again, the main endpoint here is going to be to look at safety and tolerability, but the primary endpoint is to test if there's reduced progression using a WHO related scale, and there are a whole lot of secondary endpoints. It is a double-blind, placebo-controlled trial, but it would provide a rationale for larger scale trials to look at other conditions as well. So in conclusion, the target of senolytics is senescent cells, not a single molecular pathway, as I mentioned the last time we spoke. We're targeting networks. We're not going after a single molecular target. We're going after senescent cells, in other words. intermittent treatment looks like it may be effective. In animal models, we can alleviate multiple conditions. We don't know if they work in humans. They could be dangerous in humans. Senescence is a very fundamental process. We don't know what all the downsides of clearing senescent cells are going to be. We've done a lot of work in animals to try to be as safe as possible, but our view is the only place for these agents at the moment is in carefully controlled clinical trials for serious conditions until we know if these agents are safe. We don't think they should be taken by the general public at this point or prescribed by physicians. We want to make sure they're safe, and we're trying to do this as fast as we can in multiple clinical trials in parallel instead of in series. We're not doing trials in healthy people to try to slow down processes that might take 10 or 20 years to develop. We're using these agents in conditions that are serious, that don't have good treatment options, where we know that cellular senescence is a causal contributor, where we've got good animal study data, and where we're following the patients very closely, especially for side effects. So we're trying to do these trials in parallel rather than in series to speed things up. I cannot predict at this point whether these drugs are going to work. That's why we're doing these trials. And whether I can't predict whether they're going to be safe or effective. Things often look good in mice and then turn out to be bad in people. So we've got to be very, very careful. And again, like the last time, I'll just thank a lot of people and sorry for talking so much. So I'll stop sharing at the moment.
Wow, fantastic. Thank you so much. I think, Carl mentioned in the chat, this is really a plethora of really incredible studies, and there's a lot of questions in the chat. I hope you don't mind me immediately jumping in, but this was really, really wonderful. Thank you so much. I also love the way of the presentation. I hope you don't mind if I share the slides with the rest of the group.
Sure.
Okay, good. Good. Just want to make sure. Okay, well, we have Creon's question, which got uploaded, and then we have Christine, Carl, and Alexei. Creon, go ahead and perhaps say one sentence about your background.
Oh, my background is I'm an interested amateur. I'm an applied physicist for NASA and for space companies, but I know something about molecular biology, and hence I'm going to ask you, you made this comment, mitochondrial senescent cells release mitochondrial DNA. That sounds like a pretty big deal. What the heck is going on with that? Why does that happen? Is it purely maladaptive, or is there some good reason for it? And it just looks like a serious failure inside the cell system.
Yeah, so one of the major changes that occurred, there are many changes that occur in senescent cells. There's a very complex transcription factor cascade that enforces senescence. So literally thousands of genes expression gets changed. Mitochondrial dysfunction is a hallmark of senescence. So senescent cells are resistant to dying, but they have what's called a partial Warburg shift. So much like cancer cells, they depend on sugar instead of fatty acids. There's mitochondria, a lot of turnover of mitochondrial DNA and changes in mitochondrial dynamics. So they release mitochondrial DNA, but senescent cells don't just release mitochondrial DNA, they release a lot of other non-coding nucleotides. So they release a lot of microRNAs that affect a lot of tissues at a great distance. Mitochondrial DNA is recognized as what we call a DAMP, or danger-associated molecular pattern protein. So other cells, if they detect that cells around them are breaking down or in trouble, one of their responses is to become senescent. So this is how senescence can spread, even at a great distance. And by the way, we're working with quite engaged with some of the space agencies about Senolytics for space radiation.
Yeah, because zero gravity probably introduces senescence. It introduces everything else bad.
Yeah, we've actually got cells going up on the Axiom flight in 18 months. And we're checking the astronauts on that flight.
Follow-up question, if you don't mind, based on what you said about the Warburg effect, doesn't that suggest that a very low carbohydrate diet might be senolytic?
Not necessarily senolytic as such, but there are metabolic ways of getting at senescent cells, and you do find that diet might impact senescent cell generation the same way as it does with cancers. So senescent cells have some of the same metabolic shifts as cancers.
Thanks.
So caloric restriction, for example, as I mentioned earlier, Ned Sharpless, back in 2004, published in Journal of Clinical Investigation that delays senescent cell accumulation and simultaneously improves health span.
Yeah, but it might very well be that it's not so much the caloric restriction, but the carbohydrate restriction that's stopping the sugar that they need to live.
Yeah, and we don't know that. So a lot of more work needs to be, there has been some work in that direction, but lifestyle interventions have to be looked at. But actually introducing lifestyle interventions in sick elderly people is a much taller order than small molecule pills, as much as I hate to say it. I mean, I'm a geriatrician amongst other things. I've looked after nursing home patients for many years, and I know what's possible and what isn't in some of these populations.
Thank you.
Awesome, thank you so much. I'm asking Christine Ichems 2 questions. So first of all, she says, this is fascinating. And then she wants me to ask what the difference is between senescence and terminal differentiation, and how do senolytics achieve specificity?
So terminal differentiation means the cell's acquired a specialized cell fate and stops dividing. Senescence is, in a way, a terminally differentiated state. That would be another way of looking at it. But that said, some supposedly terminally differentiated cells like neurons or like cardiac myocytes can acquire a senescence-like state, even though that's why it's really hard to define senescence, because the only definition is a normally replicating cell type that loses replicative potential. But it's beginning to look like non-replicating cell types can acquire a senescent-like state with many of the things that you see in senescent cells, including some of the organelle changes that we were talking about, changes in a lot of pathways in as far as gene and protein expression go, upregulation of anti-apoptotic pathways, and a number, many other things actually. So there's a lot of debate in the field about it, but there's a view, an emerging view, that terminally differentiated cells can acquire what looks like a senescence-related fate.
Oh, okay, thanks a lot. Next one up, we have Carl, and then Alexei, and then Jim.
So I have a couple questions, but some of them are super fast. Are you going to be doing an interim analysis at the halfway point of these trials that are at their halfway points?
Some of them, yes. Most of them, no. These are FDA regulated trials. They've got DSMBs, data safety and monitoring boards, that are following results. One of the charges, especially of NIH supported, because the NIH chooses the DSMBs for NIH trials, we don't. One of the charges in those trials is to stop the trial either if there's futility or if you demonstrate an effect that's overwhelming early.
Right, of course. If you've got good enough, the reason I asked is because if you have good enough power for an overwhelming effect, then you should see the signal. Okay, so fisetin and quercetin are not being drugs, aren't as tightly regulated in terms of manufacturing, and that's why there are companies like Consumer Lab that test them for whether, what actual level of active ingredients they have. How do you source and ensure that the doses being done in the trials are the doses you think you're giving based on labels?
We had to get a full IND for fisetin. The minute you use a natural product to treat a disease, it's considered a drug. We had to register it with the FDA as a drug. It took 2 years, 450 pages. We had to set up something called Good Manufacturing Practices manufacturing. We had to do stability testing. We had to look for degradation products. We had to test in multiple species. We had to do safety and tolerability. We had to do all of that, just like it's a brand new drug, before we were allowed to do clinical trials.
Meaning you are producing it yourself?
Yes.
Got it.
Well, we contract to a GMP manufacturer. And then
right, but you don't just get it off market. Got it. Okay.
Yeah, and then we monitor the Mayo mass spec facility monitors their product, and we have to furnish to the FDA every couple of months safety and stability data.
Understood. So wound healing, we all agree, is good, and we don't want to interfere with that. That's why we don't want to stop cells from becoming senescent. We think that most of the time when a wound happens, you get this influx of senescent cells that call this immune response that helps heal the wound, and then the body mostly cleans up the senescent cells. Do we
No, it's a
know
bit
yet?
different than that. Actually, what some senescent cells produce is platelet-derived growth factor, which helps wound healing. Those are the senescent cells that are not pro-inflammatory. Senolytics do not target those cells. So senolytics target the 30 to 70% of senescent cells that have what we call a SASP, or senescence-associated secretory phenotype. They do not target the senescent cells that produce PDGF. So we do not find, unlike what we see in animal models, if we get rid of P16 expressing cells or P21 expressing cells, we do not see the inhibition of wound healing with senolytics.
So are you saying that all the types of senescent cells that are involved in wound healing do not have SASP, or just some of them?
The SASP may be necessary early on to clear away dead tissue, as you said. But later on, it's been shown in some of the animal models where you clear cells, some of which are senescent, because neither P16 or P21 are completely sensitive or specific for senescence. But if you target P16 expressing cells and remove them, you delay wound healing. And it's been shown that if, by a group in California, that if you add back platelet-derived growth factor, you restore wound healing. And it turns out that it's the senescent cells that don't have a SASP that produce that. So you do need them early on to help clear up the wound. You wouldn't want to give it these drugs right away, but in a chronic wound, they may help.
So I guess my question is, as the immune system, as we undergo immunosenescence and maybe the cleanup is imperfect, would we expect that animals that get higher rates of idiosyncratic repeated wounds will end up with a higher senescent cell burden?
We're not sure about that, but we see that in other kinds of scenarios. So there's what we call a stairway effect. You have a burden of, you have something that causes senescent cell burden, and then you get a bit better than, but you don't get back to baseline.
I've asked a lot of questions, so I'll let other people do. Okay. And then I have
above a certain threshold, senescent cells start impeding immune system function. They produce factors that prevent macrophage diapedesis, for example. They cause fibrosis. They have don't eat me signals. They produce proteases that cleave off fast ligands. So above a certain threshold, they'll start impairing their own clearance.
Thank you. And next one up, we have Alexei. Are you here? I think you had a few questions. Alexei, if you hear, let us know. Connection's bad. Okay, then we'll go to someone else, and you let us know when the connection's better.
I have a very quick follow-up.
No, Kriyan, we're going to go with Jim O'Neill now because we have a few more people in the queue. Please just say your question in the chat.
Allison, Alexei said his connection's bad and he wants you to read his questions.
Okay, Jim, you ask your question, then I'll read Alexei's.
Yeah, you said that monthly dosing was as effective as continuous. I wonder if annual dosing would be expected to have a beneficial effect.
It depends on the rate of formation of new senescent cells, so it's condition dependent. For example, if you blast a mouse with radiation of one leg, you just need to give a couple of doses in the animal's entire life to alleviate the dysfunction. If you've got continued high-fat feeding, new senescent cells are generated at a particular rate. If you've got other stimuli that are inducing senescence, it depends on the stimulus. And it depends on the cell type that's becoming senescent and a lot of other things. So we're only just beginning to get a handle on that. And one of the things that we use the blood tests for is to try to figure out when senescent cells are coming back. We're trying to miniaturize that and make it as fast as possible so that we can do in some of these trials intermittent blood testing like every few days and determine when there's a takeoff of senescent cells again so we could dose at that point.
All right, I'm going to read out Alexei's questions now. He's asking, how much is the senescence of several layers of cells in liver an adaptive response to the aggressive environment, and how much could senolytics interfere with this response? Question number one. Question number two, how much is the senescence? Oh, no, we have, that was the first one. Maybe you just want to tackle the first one first.
Sorry, I didn't quite catch it. I heard liver, but you said something about layers.
Yeah, how much is the senescence of several layers of cells in liver an adaptive response to the aggressive environment, and how much could senolytics interfere with this response?
Well, you don't see much in the way of senescent cells in younger, healthier individuals, you know, in the liver. You see them in conditions like primary sclerosing cholangitis, where they're very abundant and they occur in one particular part of the liver around the bile ducts. There's a clinical trial envisaged in that because Nick LaRusso here has done some really great preclinical animal studies. So that'll be coming along. In the case of hepatic steatosis, where there's fat tissue that gets into the liver in the context of obesity and diabetes, there are different kinds of cells in the liver than in sclerosing cholangitis or biliary tract disease. So in diabetics, for example, or people who have obesity and hepatic steatosis and steatohepatitis, it's a different set of cells in a different part of the liver that are affected. But you tend not to see many that we're able to detect, at least using current methods, and depending on your definition, senescent cells in healthier individuals in the liver.
All right. And his second question is there any known negative interaction between the human immune system and senolytic therapy?
I think it depends on the drug. There are probably well over 100 drugs now. Some drugs are very toxic, you know, like things like navitoclax, which we in another group found was senolytic around the same time. It's a BCL2 family member inhibitor. It's used for treating, I mean, it's not an approved drug. It's an investigational drug. You know, it does have a lot of side effects on the immune system. It can cause it mainly affects platelets, but it can cause unpredictable decreases in the white count, especially of neutrophils. And after a while, there starts to be fairly severe neutropenia, where other senolytic drugs were not able to find a big effect on immune cell numbers. We find correction of immune cells in situations like coronavirus, because one of the things senescent cells produce, amongst many others that attract and anchor immune cells, is something called IP10. And it attracts certain kinds of T lymphocytes. And one of the things you see in coronavirus infection that's severe is deficiency of T lymphocytes. And you know, when we in animal models, when we treat with senolytics, we reverse that.
All right, we're now on time. There's 2 more questions. Do you have to hop off? It's totally fine.
I'm okay for a short bit, I guess.
Okay, good. Then let's see if we can make it good. I'm sorry about this bombardment, but people do have a few more questions. Joe, you are next.
Just really quickly, since I'm involved from this angle, I'm curious what you ended up paying for that GMP work, like the CMC and also the IND enabling studies for Pfizer.
Quite a lot. I can't go into that very much because I don't know the exact numbers, and we negotiated with different manufacturers, and so it depends on the study, where it is, and you know, some of the particulars of the study. The nursing home study, as you can imagine, is very complex. It's not so much the manufacturing, it's the drug delivery, which is very complex. And that was one of the big stumbling blocks to getting that trial going. So we had to figure out ways to get the drug packaged and to the nursing homes without you know, contact with people in or around the nursing homes because they're on lockdown. So it depends on the situation.
Got it. Okay.
One thing about Fizetin is we're finding it's pretty stable in our hands, you know, when we do mass spec on it. So we were worried it would break down easily. The stuff is, like, in the right storage conditions, it's remarkably stable.
Okay, one day, one day,
is that data available in terms of what the purification and stability testing and all that kind of stuff?
It will be when we start publishing some of the trials, and also the NIH with us is developing a website about that particular trial, and the website should be coming on stream in the next week or so, but it won't have the stability data and all that stuff on at the beginning, but eventually it will. It's more to share protocols and so forth and SOPs, that particular website to begin with. But we want to put everything that we can on that's not considered business confidential.
Oh, okay. Business
Fantastic.
confidential. Okay, thank you.
Well, business confidential by our lawyers. We don't, Mayo does not make money out of this.
Yeah, understood.
For
okay, thank you.
I mean, we're an academic organization, and it's,
I'm just always
and
looking
it's,
for comps.
it's government funded,
Yeah. Okay, thank you.
I'll try to look at the website and share it in the group, and if I find it.
It'll start off very small and hopefully get bigger and bigger over time.
Lovely. We have another question by Alex, and then Carl with the final word, and then I think we're good. Alex, do you want to unmute yourself?
Oh, sure. Hi, everybody. Alex Javankov. Very quick question. I'm just wondering if you have the blood tests, clinical blood tests, and the expanded biochemistry from the patients before and after senolytics, and maybe after an extended time, and if you have tried the aging clocks that utilize this kind of type of data.
Yes. The long and short answer is yes, that's all being done.
And what do you see? Do you see any?
We don't know yet. It's too, the data are just coming in and they're blinded and so forth. So we don't know. But the group at Yale, for example, that's interested in that, the epigenetics actual processing is done at University of Minnesota, and then Morgan Levine and others at Yale are looking at the results.
Are you making this data public so others can see it?
Eventually we will, yes, because these are publicly funded trials, a lot of them.
Great.
We'll have Morgan, I think, in a few months. So perhaps we can also ask her a few follow-up questions on this. And then Carl, you had a final question, and then I think we really have to.
I know Lynn also made this comment that I also made in the chat, which is kudos on just exploding so many different trials in parallel. This is great. It's great for the world. It's great for science. So I know you made a decision a long time ago when you started this kind of thing to go with systemic administration, just oral dosing, partially because of the signaling aspect, and of course we all know that Unity's local administration trial for knee arthritis failed. But given how many things you're starting, have you started to reconsider at least getting taking some portion of trials and looking at some kind of local administration? It could be the Unity's trial didn't fail because of that, but because of the chosen molecule or the indication itself, or
both. But we find there's spread of senescence. So if we transplant small numbers of senescent cells into one part of the body in experimental animals, we find spread of senescence throughout the animal. And we've got early data showing that in humans, this appears to be the case as well. So we're concerned that if there's a local accumulation of senescent cells, that may be a harbinger of a systemic problem.
Right, but
so the other issue is, how do you give things locally? For example, in a knee joint with osteoarthritis, more than half of whom, by the time they're severe enough to require a lot of intervention, don't even have a synovial membrane left anymore. If you're injecting something locally into that kind of knee joint, you're basically giving a systemic injection because the knee joint's destroyed. If you're injecting into the eye, through the canal in the optic nerve, you're delivering even large molecules into the part of the brain that controls breathing, consciousness, sleep-wake cycles, etc. You can show that antibodies injected into the eye wind up in that part of the brain very quickly. So it's really hard to know what a local injection is. I think skin is a different issue, and there's certain other things like inhaled agents. There might be a place. But it's really hard to know what you mean by local injection if there's a damaged tissue and there's a lot of blood supply to that tissue, and you could be giving at least equivalent to an intramuscular injection, if not an intravenous injection.
I suspect it especially doesn't make sense to do local when you're looking at the very, very frail, which your trials seem to be. Though as you start considering trials for younger, healthier people, possibly there are conditions where there's a very local high senescent cell burden that hasn't spread too much yet. But
well, we've got some stuff emerging from Johns Hopkins that in fact, in some younger people with local accumulations of senescent cells, they're everywhere when we look.
thanks.
Thank you so much. One final question to make those your last words. What can this group potentially do? I mean, you know a little bit who's in this group at least, to help with your work. What would you, what would be helpful?
You know, I think we've got a lot of help from the government and from foundations. I think what's very important is for people to get the message not to be taking these drugs at this point in an uncontrolled way. We don't know if they're going to be safe or effective. I give it maybe a 50-50 chance. We're doing the trials because we don't know if they're going to work. Otherwise, we wouldn't be doing trials. Yeah. You know, so as a physician, I'm very worried about the first principle in the Hippocratic Oath, you know, first do no harm. And, you know, we are playing with a very fundamental process here. And I don't know whether this is all going to work out or not. You know, humans are not big mice, and we'll have to see what happens. So I prefer sort of a conservative approach to all of this at this point. And we're trying to move quickly because we feel it's important to know one way or the other, but you know, I worry.
Yeah, you are doing the work of a lot of people, I think, in parallel at the same time. So I think we really are moving full steam ahead on this. Thank you so much for your work. Thank you so much for taking the time for sharing your work with this group. It was really fantastic. Thanks everyone for your great questions. We really appreciate it of the time that you're devoting to this group. Thank you very much. I'll be following up with a video, and if you ever want to come back on to give us an update, we would love to have you on again. Thank you from everyone.
Very much. Thanks.
And yeah, I'll be in touch. All right, everyone. It was lovely to see you all. What a full packed meeting. I'm really looking forward to our normal keynote meeting next Thursday, and it's going to be a very exciting one. Check the schedule, and I'll see you next Thursday. Have a lovely rest of the day or evening or morning. Bye, everyone.